get back into the presentations, and then we're also going to be doing, reviews for the lab exam, which is going to be on Thursday. The lab exam on Thursday will be just like the first lab exam. It'll be a proctorio administered exam, but we'll really deal with kind of more hands-on kind of questions, like pictures of what the test tubes looked like and questions about what it means or things like that. It will be, and actually when you're done with that, it's going to be kind of weird. When you're done with that, we'll be done with the class. The next time we'll all gather together is going to be Tuesday morning for the, during finals week for exam 4. What else? The optional comprehensive exam will also be available during finals week. And if you're interested in that, it can replace one of your first 3 exams. Exam 4 has to be exam 4. But if you have a grade from 1, 2, or 3 that you think, in fact, you can ask me, you can message or email me if you're wondering, like, does this even make sense? Will it make a difference? And I can look and, kind of get back and let you know if it makes sense. Obviously a major thing you want to be doing is just studying for exam 4 too, because that's a big chunk of your grade. But if you are going to take it, we'll pick a time during finals week. It can be like on Thursday or Friday even, depending on what the rest of your schedule looks like. We'll find some time that works for everybody. What else? I know that, so the lab exam is going to be open lab notebook. I've had some things happen in my life that have made it tricky to take care of stuff for the last week, and I know that the lab exam is that the labs have not been graded. They were supposed to, I was hoping by beginning of last week that it was all going to be done, and I am committing to getting it done, hopefully by the middle of tomorrow. I'm going to be working on it, all tonight and tomorrow, and I will do my very, very best to make sure that you have feedback on all of that, so you actually so it's useful. It's not going to be that useful on your lab exam if you don't even know if what you wrote is correct there. So I do apologize for that. What else? Let me go back to the modules. If we go share, the very end here, you can now see there is this research presentations discussions. So in each of these, if you here we had Sari talking last time. If you click on this, there is, I turned all of the PowerPoints into PDFs so they load much faster. So you can kind of look over the presentation and remember what it was about and all that. And part of your grade for this research project will be commenting on other people's presentations. And so I'll send it, I'll make the more formal assignment, but basically what you're going to be doing is for whichever, so I just have these in alphabetical order. For the 3, to make sure that every presentation gets the same number of comments, for the 3 presentations after yours. So if you were this one, Sari will comment on Lauren's, Tatiana's, and Wendy's presentation. And it doesn't have to be like, it can't just be totally like, great job. It should be something like, kind of engaging the material. But it doesn't have to be like, yeah, and I should also show the ground rules here. So, here's the title. And it says comments should be thoughtful, relevant, and engage the presented material. Right? So it has to actually say something meaningful. And it says you will not see your classmates' posts until you post your work. So even though there'll be at least 2 other people commenting on everyone, you're not going to see other people's comments until you put your own in, just to make sure that you don't just like write what somebody else wrote or something. You must be respectful of each other. You can challenge each other's ideas, but not each other personally. Like, this is, yeah, so, be nice. Be nice. Also, if you challenge someone's ideas, back it up with your own carefully thought out arguments, perhaps with evidence from, if you're saying like, I don't agree with these conclusions, that's part of the kind of dialogue that's actually really useful and helpful. But don't just say like, it was rigged, but not actually provide any shred of evidence or something. You need to have an actual like, I think like maybe there's more to this story and this is supported by this evidence. So if you're going to go there. Then I wrote here, remember, sometimes it's better to be kind than right. Sometimes, it's basically be nice. Don't try to trash somebody. Which I just always think is worth saying, explicitly. So, what is it? Safe. Try to make our class a safe place, to discuss ideas, challenge. I mean, in science, sometimes I find in this class people get kind of uncomfortable just being challenged, and that should be okay. It should be okay to challenge people, but it should be done in a kind way and done about the ideas and not about the person. All right, so just kind of putting that out there. Each person, and you're going to have until the end of this, till the end of finals week. This doesn't have to happen tomorrow. This can happen after you're done with exam 4 even. You can go in and start commenting on these, or you can just do it now whenever you want. But you do need to have comments for the 3 presentations that follow your own in this list. And again,
when did you say this? Sorry. When did you say this was due?
By the end of finals week.
Finally.
And I will write out the formal assignment so you have it written out after class today. All right. So the fact is, we are not going to have time for everybody to do a formal presentation because of the time, I'm realizing. So we have time for a number of them, and I can just put it out there for if there's people who particularly would like to present and share, you can let me know right now. Just type it in the chat or just say it out loud, or else I will randomly choose people. Kimberly. So Kimberly, Diana Pineda, and Sunny. So we have, in Kimberly, do you go by Kimberly or Diana?
Either one is fine.
Okay. All right, so I'm going to stop sharing here. And do you want me to present your thing, or can you share?
I can share it.
Okay, so let us, and again, like last class, you know, please give our presenters your full attention, and let's hand over the Zoom room to Kimberly and Sunny.
Okay. Can you see the presentation?
Yeah.
You want to put it in full screen? Yeah.
Yeah, there you go. So first of all, I apologize for the background noise. We have some maintenance going on in the back. So, okay, let's begin. So Sunny and I are going to discuss alcohol consumption during pregnancy. And we're going to start by explaining what is alcohol. Alcohol, it's a teratogen. It's a factor that causes abnormalities and malformations of the embryo. It's also known as ethanol, ethyl alcohol, or grain alcohol. It's a natural byproduct of plant fermentation, and it's the main ingredient in alcoholic beverages such as beer, rum, vodka, etc. Some of the physical properties of ethanol include clear color, liquid. It's unable to dissolve in fats and oils, and it can pass through membranes of our body. So how does the body distribute or break down alcohol? So alcohol has different effects on different people, and it depends on the blood alcohol concentration of each person. After alcohol is swallowed, it's mostly absorbed in the stomach, and then it goes through the small intestine into the portal veins of the portal system, which leads to the liver. Alcohol also gets broken down by certain enzymes in the stomach and liver. I can't even pronounce them, so they're right here on the PowerPoint for you. Also, the brain has something to do with alcohol breakdown. Ethanol travels from the blood and distributes into all the surrounding tissues and fluids in proportion to the relative content of water, which can be explained by the process of passive diffusion when things are moving against concentration gradient. So next, why is alcohol bad? Well, we know the effects of alcohol, but definitely alcohol has terrible effects on the brain. And some of this include like certain behaviors most of us have experienced it if we've drank alcohol, slur speech, poor memory, slow reflexes. The brain is also responsible, like I said, for certain breakdown of the alcohol, and it disrupts communication pathways and blocks chemical signals between cells in the brain. So accumulated evidence in several rodents have shown that ethanol treatment produces cognitive impairment in the hippocampus. We know the hippocampus plays an important role in memory formation, emotion processing, and learning. So deterioration of the hippocampus results in difficulties or in lack of cognitive skills. The hippocampus, as we know, home of many neuron cells and neuron processes, which is how the brain receives and breaks down information from incoming synapses. So damage to the hippocampus, again, results in neural cell loss and also contributes to body inability to carry out corresponding functions to incoming messages from the synapses through the dendrites. It can also damage the dendrites or change it. So alcohol and pregnancy. During pregnancy, there's a developing baby in the woman's womb, causing the alcohol to take a detour and pass through the umbilical cord into the placenta and making its way to the fetal maternal sections. Alcohol then diffuses across the membranes, making sure that alcohol consumption stays the same between the mother and the fetus. Unfortunately, the fetus doesn't have the enzymes to break down alcohol, and it's unable to metabolize it. So the blood alcohol concentration of the mother and the baby are at the same level, which is really, really bad for the baby. Next, I want to talk about what causes women to drink during pregnancy. And there are different reasons why women drink during pregnancy. The result of some findings show that most of the time the mother was an alcoholic before conception. However, recent studies have shown that between 56% and 92% of alcohol users have other psychiatric illnesses, and up to 70% have experienced childhood abuse. Women may use alcohol as coping mechanisms to deal with difficult situations in life. Some factors that were determined to increase the risk of women gaining alcohol dependency, including poverty, homelessness, substance abuse of their partners, or post-traumatic experiences. In some cases, women's alcohol dependency from preconception motivated her to ignore her pregnancy so she can continue to drink. This is considered to be a judgment-impaired decision, which is often a symptom of excessive alcohol consumption resulting from the damages happening in her brain.
Okay, I'm gonna kind of building off of what we were talking about earlier, that alcohol diffuses through the fetal maternal membrane. So the fetus, since it's unable to break down the alcohol, that means that the ethanol stays in the fetus's system way longer because it doesn't know where to go. And so the effects of alcohol on a fetus depend not only on the amount of alcohol that's consumed by the mother, but also at what stage in development or during their pregnancy they drank it in. You want to? Okay, here you go. So, like I said, alcohol passes through the umbilical cord into the placenta and into the baby's bloodstream. And what we know is that the umbilical cord and the placenta are it's the main source of how the fetus is getting its nutrients, the blood, and how it's building. So basically, whatever the mother is consuming, it always has an effect on the child or the fetus. And alcohol ends up causing many birth defects because it disrupts the developmental process. Some of the more obvious birth defects are physical abnormalities. And later we'll talk about fetal alcohol syndrome, which is directly associated with maternal alcohol consumption. And it could lead to mental retardation. These effects, they continue to impact the child throughout, and they're irreversible. So there's no turning back from it. There's no way to fix it. There's no way there, at least not yet, that after the child is born with these abnormalities, that doctors could go back and fix it. It's not just something as simple as like a cleft lip or like a messed up eye. It's multiple things and a lot of effects on the brain. Another thing that we'll talk about is sudden infant death syndrome, which has been associated with fetal alcohol, maternal consumption of alcohol. So as we know, when during pregnancy, the fetus develops over a period of 9 months. And so during the embryonic stage, the body plan is laid out in which it will turn into the organ systems. So when a woman drinks alcohol during the embryonic stage, it can really mess up how the blastocyte is placed to even begin fertilization. So even before fertilization begins, it could mess up the process of even getting pregnant. In the third or fourth week, in this first or second week, when fertilization begins, like I said, it has the misplacement of blastocyte. And for women that drink a lot of excessive alcohol, it could terminate the pregnancy completely by causing a miscarriage. In the third or fourth week, we see that the fetus heartbeat is typically detected. And this means that if the alcohol is consumed, then it could mess up the process of the heart formation, and it could mess up how the valves are made, and just in general heart problems. And during the third to sixth week, the embryo starts to form the neural tube and cranial bones. And this is what we saw in almost every case, is that it affected the brain somehow. And earlier, like we were explaining, alcohol has severe effects on the adult brain. So if it has those severe effects on the adult brain, I mean, we can just like imagine and think about it, like what kind of effects it would have on a brain that's not even developed yet, or how it could mess up that mess up that process. In the sixth and seventh week, the brain becomes more detailed. So it starts to become into different parts. And like we were talking about the hippocampus, a lot of researchers show that it directly affected the hippocampus of the child, which explains like why there's behavioral issues, mental retardation, their social cues could be off, and just the learning processes could be slowed. I mean, it shows up in so many different aspects. So the two most common outcomes on the fetus after the fetus was born out of maternal alcohol consumption is fetal alcohol syndrome and sudden infant death syndrome. And so fetal alcohol syndrome is any change or condition in a child that directly resulted from maternal alcohol consumption. And this one, it was more physical. It was more of they talked of more physical appearances, but it directly related to psychological issues too, because of the disruption in the brain formation. And so some of the physical impairments included eye abnormalities, eyelid openings, skin folds on the ear or the eyes. The eyes over here, it could be smaller or larger, or they could even be droopy. SIDS is the sudden unexplained death of an infant under 1 year of age. And so a lot of studies show that the mothers who drank excessive alcohol during the first trimester of pregnancy, they were more likely to have a child and the child die from sudden infant death syndrome. And there's really it happens very sudden. It literally is sudden infant death syndrome. Like it often happens in the sleep. It doesn't even matter how the baby is sleeping, they just pass away. And so we looked a lot as to maybe like what different sources there are to help women who have addiction and who want to stop. And we found these alarming statistics, but we did want to show you guys that it's they say not to drink alcohol at all, like to completely stop it as soon as you know you're pregnant. Like if you knew you're pregnant and you drink it, your child's life is just going to be ruined completely. And so these are two pictures of abnormalities that could happen. If you could see on this kind of cartoonish picture, the eyelids are very, there's basically no eyelids. The top of it is very down. And the eyebrows are also very uneven. They could have a thin upper lip, but they could also have like abnormalities in their lips as well. And then just their facial bones, it really affects their face. From what we are seeing is that their face and their brain is severely impacted. And so this is a picture of a real child, and as you can see, they're very similar findings. So we would advise not drinking when you're pregnant because it's irreversible and it affects the child forever.
All right, thank you. All right, does people have any questions for them? I think one of the things that's kind of scary about this is that those real like critical embryological development, people don't even know they're pregnant yet. That's before you even know like you've missed your period. Some of those real critical like organogenesis things are just starting to begin. So it's definitely, even if you're trying to get pregnant, you probably want to be careful about alcohol. And it is it does, from at least the stuff I've looked at, it does seem that it's someone who's pregnant and had a glass of wine, it's very likely didn't affect anything, but there are these critical periods where particularly a large amount at a critical point in development can have have a large effect. So it's kind of non-linear. Um, so yeah, no, but it's scary. Cool, thank you. All right, so Wendy asked to go next. So let's see. Let's give our hand over the Zoom room to Wendy Mazariegos.
Thank you.
Sure.
All righty. So just press share screen.
Exactly.
Okay. Oh, geez. Okay, can you guys see it?
Yeah, it looks good.
Okay,
Yep.
thank you. Alrighty. So I am doing, or I wrote my paper and did my presentation on using LSD as a treatment for mental health issues. As an introduction and background, LSD, or sorry, I'm gonna botch it, lysergic acid diethylamide, is a synthetic chemical that was created by Albert Hofmann. He's a Swiss chemist, and he discovered it in 1938. It was, however, until 1943 when Albert Hofmann discovered its hallucinogenic effects. So I was reading a lot of articles and it said like he tried it by accident or just, or he might have tried it on purpose. I don't know. Either way, he discovered a lot. So today LSD is listed by the DEA as a Schedule I substance because of its potential to be abused. So it's pretty high up there with heroin. How LSD works on the brain. So LSD as a chemical will bind to dopamine, adrenergic, serotonin, and glutamate receptors. The most talked about receptor, however, is the 5-HT2A receptor. And it's claimed to be the site where the hallucinogenic properties are produced. As we learned in lecture, LSD is a promiscuous ligand and will act as a partial agonist to the 5-HT2A receptor. So it means that it will bind to it, but it'll also create change or activate the receptor. And okay, so I was interested in this topic as I myself have struggled with generalized anxiety since I was very young. I've tried cognitive therapy, talk therapy, a lot of medications, and medications can be a little tough to take, especially if you're in school. So I don't know, I was just naturally interested in this kind of controversial treatment. So let me see. So I also found a lot of articles about psychedelics making headlines in the last several years and becoming more accepted as a potential anxiety and depression treatment. So I also wanted to find out about the efficacy of this method. As statistics show, these are percentages by the CDC. Just this year, 11.2% adults reported feeling just general anxiety or worry, and 4.7% experienced depression. Sadly, I think this number would be a lot higher if it included preteens and teenagers. But that data isn't in that because this is over age 18. So the amygdala and LSD. As we learned in class, the structure is what helps us process our fight or flight response. As a recent study from 2017 found that LSD in a 200 microgram dose has the capability to reduce activity in the amygdala when introduced to fearful stimuli. They used fMRI imaging to see these changes. Additionally, LSD is reported to increase trust, which I thought could be helpful for relationships with therapists. If you're taking it, and also, I don't know if it helps build trust with your therapist and open up more and create change that might be helpful. There's a lot of studies on that too. And fear perception and LSD. So in another 2016 study, researchers asked their participants to rate neutral emotional faces as happy, angry, or fearful. Both the participants that took the 100 microdose and 200 microdose had a fear recognition impairment and were less likely to rate fearful faces, were less likely to perceive fear in those faces that were actually supposed to be rated as fear. This shows how LSD might change the way that we perceive and process our emotions and how we process threat. Anxiety in cancer patients. Throughout my research, I found many participants were cancer patients or patients suffering from terminal illness anxiety. This team of researchers, Gasser et al. In 2014 tested 11 patients and analyzed their anxiety levels with this intense scale. I don't know, it's called the STAI scale, the Spielberger State Trait Anxiety Inventory. So 8 patients received the 200 microdose and 4 initially got the 20 microdose, microgram dose. And then once they realized that the patients that got the 20 microgram dose weren't having any improvement, they would switch them to the 200 micrograms. And it was reported that at 2 months post-treatment, all patients' anxiety had decreased relative to their baseline levels. And that 12 months post-treatment, the scale revealed the anxiety reduction levels sustained. So through those 12 months, those patients had still had the same anxiety level as the second month. So that could show a little bit about long-term and how this could have a benefit. So are adverse effects. So obviously there can be impairment in how a person navigates their world while taking this chemical. It can raise blood pressure, heart rate, dilate pupils, um, and then of course cause visual hallucinations. Most concerning is that there have been reports of some people having anxiety or panic attacks, which is what they're trying to help. So that would be bad. That's a big no-no. So that would be called a bad trip in layman terms. So this is why more research, I think, is needed to see what a safe dose is and what mindset a patient should be in before taking this therapy as a therapy. So psychedelics long-term. In 2015, Peter Hendricks and his team found that lifelong psychedelic users actually reported reduced stress, reduced suicidal thinking, and reduced suicide attempts when other drugs actually increase these outcomes. So this raises the question of how often would a patient need to take this form of therapy? And also, it's a good note that this study that I just talked about, the psychedelic users, they were taking psychedelics like psilocybin. Sorry if I can't pronounce it correctly. And that drugs like, or substances like psilocybin are more accepted because they have less intense effects. And they're just more accepted in the research community. So as a patient, what would I want to know? I would want to know what the safe dosage is, frequency of taking LSD as a therapy. Would taking this cause any damage to my organs? Do I have to get myself in a right mindset before taking it? And perhaps this therapy would help those with, would mainly only help those with end of life anxiety. And if taking it, maybe there would have to be a controlled area, controlled clinic. And yeah, in conclusion, LSD shows both positives and negatives. I personally don't know if I support it, but think increasing research is important. In November, I don't know if you guys don't know, but Oregon became the first state to legalize psychedelic mushrooms in 2020. So you can see how there's more acceptance in both the society, research, and medical community. So thank you very much. That's my presentation.
Thanks. All right, so does anyone have questions for Wendy? I'm surprised that she brought up a lot of interesting stuff there. Yeah, I mean and I should mention too when she's talking about 200 mics, that's solid. Like these people are not taking something that they're kind of feeling. 200 micrograms is very deep in the experience, and yeah, definitely the whole idea of set and setting is really important. Like at that level, if you're not in a safe place or your intentions are not clear about what's going on, it can be pretty traumatizing. Or, people also have amazingly beautiful spiritual experiences, but it's a heavy, what they're experiencing is, it's not like taking Prozac or benzos and just feeling less stress. I mean, there's also a lot of question about how much of the effect, like at that dose, the effects seem to be more about like what you're experiencing, like in how you're processing through things that you're holding, as opposed to like the microdosing, which people also has gotten very popular, which is something very different. That is taking much smaller amounts that seem to be more kind of just chronically adjusting your neurochemistry. But the dosages that Wendy was talking about there, that's a heavy trip. But yeah, people are doing a lot of research with LSD, and psilocybin and LSD have very kind of similar abilities to take you into all those different places, as opposed to like the MDMA work, which is pretty. It's a fundamentally different. It's kind of what they call more of an empathogen. Like it, some of the people who have worked, have been involved in this research, like the idea that like these more true psychedelics allow people to have a more dramatic ability to go into lots of different spaces, but then that can also be tricky. MDMA work tends to be a little more controlled because it tends to not allow quite going into the hell realms as easily. um, what else I should
So
mention?
when those people are like on a bad trip, or if it goes bad and they're like in a controlled environment, is there a way to like reverse it, or do they have to just like wait with them through it and like help them get through it?
It's more of that. It's helping them get through it. And and the idea of what is a bad trip is it's kind of a misnomer. If you're doing it with an intention and in a controlled, safe environment, sometimes it's a very excruciating and unpleasant experience, but it's ultimately working to kind of integrate something that ultimately is going to be helpful. So so sometimes, yeah, a lot of the really transformative experiences are very, very um, excruciatingly painful or sad or whatever. But that is ultimately not a bad thing. So,
Besides the potential for abuse, are there like any other potential negative effects that have been studied or looked
yeah,
at?
so there's very little potential. I mean, one of the weirdnesses about it being Scheduled 1 is there's very little schedule for or potential for abuse with the psychedelics because you don't have a like repeatable or like a positive experience that you can count on. If you're just like taking a psychedelic to like get off, you could end up having a horrible time. It's not like if you're shooting up opiates, you know that it's going to float you someplace nice. Like opiates have a really high, or amphetamines. Like you're eating your amphetamines and you're just gonna or cocaine things. A lot of those really do give you a repeatable, kind of reliable, positive experience. But psychedelics don't typically do that, so it's very rare for people to get into that kind of relationship with them.
Yeah, if you really look into the articles and um, just the presentations of it, there's a connections to like like how the FBI controlled it and did all these projects with it on people without their consent, and it gets kind of creepy.
Yes, yeah, it's weird. So, and I will say, like, just a little story about Albert Hoffman is actually interesting. He was working for Sandoz, he was a natural products chemist, and he was working on taking the products of this ergot fungus and changing the molecules slightly this way and this way and this way and this way and this way, and seeing if it would end up being a useful drug, and came up with drugs that were useful for migraines and things like that. And the story goes, at least, is like he just had this like LSD 25. It was like the 25th um, drug in this series he had manufactured, and he had this idea like, I think it's useful. And like he made some, and he did he self-administered. Back then, that was one of the ways you could check if something was useful. And you start with a super little dose, and then you can see, like, if it's not healthy, I can feel this isn't good, or if it's interesting, I can ramp it up. And so he took 200 micrograms, like a fifth of a milligram, which for almost any drug would not be, would be below where it would actually hit you. But for acid, it is really heavy. And so he took it and he was biking home, and all of a sudden everything started melting and getting weird. And he was worried he was dying. He called he had like a doctor come over and kind of check him, and once he knew that he was like actually physically safe, I think he drank some milk and like had a kind of pretty amazing time on it. But they still they call it Bicycle Day. Like April 19th, 1943 was the first like intentional LSD trip, and they're still like people get together in Golden Gate Park and celebrate Bicycle Day every year.
That's why I kept seeing bicycle pictures when I Googled all these images. I was like, what is up with this bicycle stuff?
Because that was the first intentional like heavy acid trip, was him like biking home from work in Switzerland from his office back to his home, not expecting anything crazy like that was going to happen to him. So yeah, it was pretty intense. All right, we think we have time for one last one before we shift gears here. So, does anybody want to? Oh, I see Jamba Lama is going to now take over the floor and
Okay, I'll just keep it really, really short.
yeah, so yeah,
Uh,
definitely if you could, that would be good.
and let's see if I can share my screen.
And if you're having trouble, I can always do it for you as well.
Um,
There you
can you
go.
see it?
Yep, you're good.
Okay, it's pretty rough. I didn't prepare it nicely, but just enough so everybody can see. Yeah.
Well, actually, let me. I'm gonna do it because yours it is kind of hard to see there. So hold on. Um,
I'm gonna go really fast. So can I just control it?
go for it.
Okay, thank you. So hi, my name is Jamba, and I'm going to be talking about the type 2 diabetes and its association with the heavy rice consumption. Um, so more than 34 million Americans have diabetes, and 95% of them have the type 2 diabetes. Type 2 diabetes often develops in older age people, but now we're seeing the disease indiscriminately attacking people of all ages. More and more younger people are also developing it. What causes type 2 diabetes? Type 2 diabetes occurs when your body cell doesn't respond to insulin hormone released by pancreatic islet beta cells. Insulin works like a key to let glucose into the cells. Cells use glucose to make ATP via cellular respiration. If you have a type 2 diabetes, cells don't respond normally to insulin. As a result, the more you eat, the more glucose is produced, and the more it accumulates in the blood, setting the stage for hyperglycemia, which means you have a lot of glucose in your blood. Pancreas respond to hyperglycemia by making more insulin to try to get cells to respond. Eventually, your pancreas can't keep up and your blood sugar spikes, setting the stage for prediabetes and type 2 diabetes. High blood sugar is damaging to the body and can cause other serious health problems such as heart disease, vision loss, and kidney disease. So this next one is how the insulin is released from the beta cells. So beta cells are the ones which is making the insulin in the pancreas. So what happens is when your glucose level rises up, it enters the beta cells uh, through a glucose transport chain, the door, and once it's in the beta cells, it goes through glycolysis, the cellular respiration to make it ATPs. So when there's enough ATPs generated inside the beta cells, it causes the potassium channels to close and creates a potassium gradient inside the beta cell, which then depolarizes the calcium channel, and it opens It's stored for a calcium channel, calcium ion to flow into the beta cells. So this causes the exocytosis of the insulin out of the beta cells. So clinical manifestation of type 2 diabetes are one of them is glycosuria, which means that you have a lot of sugar in your urine. So when that happens, because glucose is osmotically very active, it pulls water with it in the urine. So when that happens, you get polyuria, which means that you're peeing a lot in your urine. So when that happens, you lose a lot of blood volume, and your blood becomes hyperosmolar blood. And when that happens, you get this thing called polydipsia, which means that you start to feel more thirsty. So, and that happens when the hyperosmolar blood signal, well, the brain hypothalamus gets a signal from the blood, and it triggers the thirst. So next thing, what happens is the polyphagia. It's the you get hungry more often, and that happens because your cell can't utilize the glucose to make an ATP. It starts tapping into the protein and fat in your muscles and other parts of your body, and starts to generate ATP through that instead of glucose. So when that happens, you start to experience a weight loss. So when you are experiencing weight loss, your brain will tell you, your hypothalamus will tell you, okay, you should eat more because you're losing a weight, and you get this thing called polyphagia because you start to get really hungry, and you start to eat more and more because you're losing a lot of weight. The next thing is the chronic complication associated with diabetes. So when there's a high level of glucose, they like to bind to protein with this non-enzymatic glycation, which actually forms a very potent inflammatory molecule that causes atherosclerosis and hyaline arteriolosclerosis in the blood vessels, which is like a plaque in your blood vessels, which impedes blood flow and proper gas exchange and nutrient exchange. So when that happens, it develops into a coronary artery diseases in the heart, and you get myocardial infarctions. And if it happens, in your lower extremities, like in your foot, you get a foot ulcer because not enough blood is flowing to your foot, and whatever is flowing down there is like mostly a sugar, and it doesn't help. Another thing that happens is that you get ischemic stroke. The less blood is when less blood flows through to your head, then you start to experience this ischemic stroke. And another effects of the atherosclerosis is that it affects your vision too. It starts to form like a microaneurysm, the cottonwool spots, flame hemorrhage in the eye, the blood vessels of the eye, and which all affects the vision. Another thing that caused by the too much sugar in your blood is hyaline arteriolosclerosis, which is the also a type of plaque which deposits in your glomeruli, and this causes albuminuria, which is like I start to lose albumin protein in urine. So when this happens, you get chronic kidney diseases. Next one. What causes insulin resistance? Factors such as genetics, family history of type 2 diabetes mellitus, obesity, and old age all have been shown to increase the risk of insulin resistance. Heavy consumption of white rice have also been associated with the insulin resistance because of its high glycemic index that can spike your blood glucose level quickly. More than half of the world's population heavily relies on the rice because it is cheap, has a long shelf life, and provides a good source of energy to do heavy work. Asia is the largest producer and the largest consumer of the white rice. Because white rice is heavily consumed in Asian countries, type 2 diabetes is more prevalent among Asian populations than other people around the world. White rice is the most common type of rice which has been highly processed. Due to heavy refining, most of the essential nutrients are lost with the outer germ and the bran layer. The remaining endosperm is mostly starch. The exposed starch can be rapidly digested, absorbed, metabolized by the body into a sugar, which increases blood sugar level quickly. Consuming too much of the highly processed white rice on a regular basis can increase the risk of type 2 diabetes. So this is the picture of the rice grain, and you know, the one in the middle, the tiny part, the endosperm, is the white rice, and the other outer two parts are heavily refined and they're taken out. So the only thing that you are getting from white rice is this little tiny middle part, which is all starch, carbohydrates. So the picture shows like the people of different continents eat how much rice. Like you can, like here you see in Asia, they eat like 10 times more than people in North America right here. And you can all see the difference, how much rice they eat. Research conducted by Keeson in 2010 that consisted of 39,765 men and 157,463 women participants found 16% lower risk of type 2 diabetes in participants who substituted white rice for less processed brown rice, and 36% decreased risk of type 2 diabetes in another group of participants who replaced their white rice diet with whole grain rice. Arsenic contamination in rice is also linked to prevalence of type 2 diabetes in South Asia. Inorganic arsenic is a carcinogen naturally occurring heavy metal found in soil and drinking water globally. However, much higher concentration of arsenic has been reported in the underground water of South Asian countries, which is the major source of water for agricultural use, as well as drinking water. According to Wao Xing in 2008 journal article, rice plants absorbs more organic arsenic from the soil compared to other plants. The study's results showed that brown rice contains 80% more inorganic arsenic than white rice because considerable amount of arsenic is found to be present in the germ layer of the brown rice. But much lower level of arsenic contamination has been observed in white rice because the germ layer is removed during heavy refining. There are many strong compounding evidences that heavy rice consumption is strongly associated to type 2 diabetes because of high glycemic index and arsenic contamination. Studies also show Asians are more prone to getting the disease because of a genetic predisposition and heavy rice consumption. Cases of type 2 diabetes have quadrupled globally, with 90% cases occurring mostly in India and China, the two most populated countries in the world. Besides being the highest consumer of rice in the world, which many studies have claimed to be strongly associated to increased risk of type 2 diabetes, sedentary lifestyle, unhealthy eating, as well as genetic predisposition are the key driver of type 2 diabetes global epidemic. And that's all I have for presentation. And thank you for listening.
All right, thank you. All right, So do we have any questions for Jamba?
It was very interesting, Jamba. I love rice actually. And I didn't know that it had a high glycemic index. I should have, but I didn't. So it was very interesting, and mostly that, of course, in countries like Asia that has the highest consumption, I didn't know that the prevalence of diabetes was so high. So it was completely new to me. Cool.
So, I mean,
Yeah, I mean, I'm actually from that country. I'm from Asia, from Nepal, and people there eat so much rice. It's just if people here see how much people they eat there over there eat rice, they'll be surprised. It's like they eat the whole full plate of rice like three times as like 3 or 4 times each day, and it's
In my country too, because it's cheap.
yeah, it's very cheap and yeah, it's available everywhere, and people are hooked to it. You know, like people feed like even the kids. They have been trained to eat rice from the childhood, and it's like when they grow up, they can't stop eating rice because it has grown into their uh, behavior, and it's really hard to change that habit. So it's almost like they can't live without rice, just like I think like some people who can't live without burgers here, I think.
I mean, what we have in we like like in the U.S., like white flour is similar, and white flour is basically taking the grain from the wheat and getting rid of the germ and bran and all of the parts with nutrients and everything, and just basically having the starch left over. Which again is going to be kind of pure carbohydrate, and then without the nutrients, like enriched flour. I mean, when I first taught nutrition, I hadn't really thought about it much, and I had to like look into this deeper, and realizing what we do is we take the seed, we rub off all of the good stuff with the nutrients on it till we just end up with some starch, and then we add in some vitamins that so it at least has some of the stuff that we got rid of that would have just been there with so much more if we had just used whole grain. So which is yeah, but that's kind of the way we do it, which is kind of fascinating. I mean, so you said that the incidence of type 2 diabetes is quadrupling. Is like the consumption of rice growing, or do you think it's other factors that are figuring into
Well, in Asia, particularly, the incidence type 2 diabetes is increasing because of the heavy rice consumption. That was the main association in type 2 diabetes and the rice consumption.
the high what?
Um, also, I heard that Asians are more prone to getting diabetes, type 2 diabetes. That it has to do with the genes, and it's that area of the genes. It's still there are so many studies that people are still finding what's causing uh, it. Uh, what is happening in the genes that's causing more diabetes among Asians. Um, I really don't
Okay,
know much about it, but
I just kind of curious.
I'm just putting it out there.
All
Uh,
right, so what we're gonna do now
um,
is we're gonna shift gears, and we are gonna spend a little bit of time just talking about the lab exam coming up. So, for the lab exam, like I said, it's going to be based on all of your labs. Oops, hold on. If I share, it's going to be starting with like, rods and cones lab onward. You know, you still need to remember the bigger concepts like independent and dependent variables or qualitative versus quantitative data and things like that. But the focus is going to be on the labs that we did, you know, post the sensory labs. So starting with the rods and cones lab. You know, and again, for each lab, it's, if relevant, it's going to be important to remember what were the independent versus dependent variables. You know, how did we go about actually measuring things? You know, what did we actually find? You know, so for rods and cones lab, if we just start there, you know, what was the question we were asking? What was the objective there?
The relationship between light intensity and the speed of the visual reflex.
Exactly. We had the independent variable was varying the intensity of the light. Dependent variable was measuring the speed of this visual reflex, like how fast you could grab the stick once you noticed you could see it falling. Um, what did we find?
The dimmer it got, the slower the reflex was.
And why did that make sense?
Because the rods are taking, you're using your rod vision, and those are slower than cones.
Yeah, so in the dimmer light, we used rods. So, and how did we control for just like making sure that people like, because we know that just doing some task, if you're doing the same thing again and again, you usually get better at it. How did we make sure that kind of practicing was not affecting our results?
Randomized trial order.
Say it again, Lauren.
Randomized trial order.
And
Like randomizing the light intensity.
yeah, exactly. So we had made sure that the intensity of the light and the order of the trials, so which would be correlated with practice, were going to be dissociated or disentangled from each other. And again, that's something, we didn't actually design our own experiments this semester because of the pandemic. But when you're designing your own experiments, that is something you really need to think about, is kind of disentangling potential sources of variability so you can analyze your data in such a way to definitively say, we could graph our data like, speed of the reflex versus light or speed of the reflex versus practice, and they were, they were disentangled because of that randomization. So that's important when you're thinking about just designing experiments. I think I'm looking here. Well, then we got all into all the cardiovascular ones. Here we had 3 different labs. We had the one that was about blood pressure. We had the one that was about the electrocardiogram. We had one that was about blood work. And we also had the shorter one, which was kind of pulses and heart sounds. These were not so much about hypothesis driven. These were more kind of just exploring these different aspects of the cardiovascular system. For each of these, for blood pressure, we did 2 big things. One was we talked about just how you actually measure blood pressure using that auscultatory method. When you first put the stethoscope on the person's brachial artery and are listening, and you pump the cuff all the way up to like 180 or whatever, what are you going to hear?
Nothing.
Nothing. And how come you're not going to hear anything?
Because the vessels are constricted, there's no blood flow.
Exactly. As long as the pressure in the cuff is greater than your systolic, no blood's moving down the arm, so you don't hear anything. What happens as you start lowering the pressure in the cuff?
Turbulent flow.
Yeah, at some point. When did that turbulent flow start?
Once the cuff matches the systolic pressure of the artery.
Or probably once it drops below. When it matches, it's just there. As soon as it drops below, you're going to start having some blood squishing under, and then you'll hear it. Because again, I talked about smooth flow, you don't hear like, but turbulent flow, you hear if there's a flow that's going all turbulent. Eventually, the sounds of cough stop as you keep letting the pressure out. And what does that tell you?
There's just natural flow and no longer constriction in the vessels.
And what is the significance of that moment when you notice that pressure?
That it's lower than the diastolic.
Yeah, exactly. When you're lower than the diastolic, that's when you're going to finally not have any more constriction. It'll start just kind of flowing more normally. So, and we looked at blood pressure happening in different situations. Like, what happens to your blood pressure and pulse if you're exercising?
Increases.
It increases. Which makes sense. If you have increased need for oxygen because either going to your muscles or kind of recovering from oxygen debt, you're going to need increased flow, which is going to need increased pressure. Do both systolic, diastolic increase similarly?
No, the diastolic doesn't, because that would be counterintuitive, because that keeps the blood from ejecting from the ventricles.
Right. So we saw there's a pretty dramatic difference in what was happening there. We also looked at the blood pressure in response to pain. It's like with the cold pressor test. What happens to like your blood pressure and pulse when you're in pain?
It varied.
We saw this pretty big variability, like the hyporeactors, the hyperreactors. That's something, again, when I think about things that I would like you to walk away from this semester with, that's another important thing about how sometimes your just intuition about what's obvious is not necessarily true, that there's a lot of these idiosyncratic responses where one individual is going to respond very differently from another individual, and it's just the way it is. We're all put together differently. There's all these genetic polymorphisms that create this very unique set of cells, and everybody's body is a little different. Everybody's responses are usually a little different. There are in general often trends, but there's also a lot of variability. We looked at blood pressure and smoking. What's the typical response of your pulse and blood pressure if you're taking a toke on tobacco?
Increases blood pressure.
Why would it increase blood pressure? At least acutely.
Vasoconstriction.
Vasoconstriction, nicotine is vasoconstriction.
For constricting the vessels, and we've seen that, back in classic thing that increases blood pressure, vasoconstriction. For blood work, you need to make sure you know about the different blood types, how we identified them, who can donate to who can receive from who. I happen to be A positive. Who can I donate my blood to?
A.
Positive and AB.
I can give it to other A's.
And AB.
and I can give it to AB, who is the universal recipient. And since I'm positive, I would probably give it to somebody who's positive, or else they could get it the first time, assuming they've never seen it, but then they would have a response. If I was type AB, who could I give blood to?
Only AB.
Only AB. Anybody else is going to react against the A marker or the B marker. All right, so make sure that that all makes sense to you. What do we call it if we take your blood and put it in a little tube and just spin it down and look at the ratio of cells to the overall blood volume?
Hematocrit.
Hematocrit. Right, so what's a typical hematocrit?
55-45.
55 would be high, like 40, 45.
40,
Up to
45. Okay.
50. A guy could get up to 50 or a little above. 55 would be getting to the top range for sure. What else? We talked about coagulation time, and that's actually important. How did we test for coagulation time?
Broke the little non-heparinized capsules and checked time intervals until we saw like the little fibrous formations.
Yes, we used those little capillary tubes. We had to make sure it was non-heparinized, right? Because for the hematocrit, you actually need the heparin to make sure it doesn't clot up on you. But if you're going to actually want the blood to coagulate, you're going to have to use the other ones to allow it to happen. And for our lab, what did we see could affect coagulation time?
Aspirin.
Aspirin. I mean, by kind of dramatic, kind of like doubled it. And like I said, sometimes just some herbal things that people take, like ginkgo biloba, are pretty dramatic at increasing the coagulation time. And again, a lot of drugs that are taken on purpose to try to prevent aberrant blood clots, like Coumadin, warfarin, increase, make it less likely that the blood's going to clot. Like, so if you're worried that there's a danger that there's going to be an aberrant blood clot, a little thrombus that's going to break free and cause a stroke or something, then, the person takes this drug, which makes it less likely for that blood clot to happen. What's the danger of overdoing it with one of these these drugs?
Bleed out.
What's that?
You would bleed out.
Yeah, exactly. It means then you're kind of like hemophilia, right? Then you just don't stop bleeding. You need that. You're always kind of breaking little blood vessels. People who have too much of these blood thinners on board are going to just get horrible bruises just from trying to put on their socks or something. What else? Pulse and heart sounds. Everybody hopefully spent some time feeling what the pulses felt like. Some are more tricky to find than others. What kind of affects how strong the pulse feels when you try to feel for a pulse?
How deep the artery is and if there's bone underneath.
Yeah, exactly. That's a big part. Like if it's too deep, then you're not going to feel it. Like your aorta is the biggest artery of all, but it's just buried deep in your chest, so you don't feel that one. Or if there's nothing underneath it, you push on it, but it just runs away from you, so you're not going to feel it. You feel it much more if you can kind of pin it between some underlying bone as it's pulsing. The radial artery is a great one for that. You push it down, it's sitting on, remember from anatomy, the bottom of the radius is this big flat triangle, and you're just shoving the artery down on top of it, and it's like pushing up at you. So that one's like particularly kind of, easy to find. But again, it's worth thinking about pulses because they're used diagnostically a lot, just to get a sense if there's adequate blood flow coming down to different parts of the body, as well as for checking out your heart rate and things like that. And again, for pressure points, knowing where to push. We saw in the EKG lab that, by knowing where the brachial artery is and pushing, you can basically stop the blood flow down to your hands. So if somebody is hemorrhaging really bad from some bad injury, knowing where those main arteries are and pushing on those pressure points can be really valuable. EKG, what is the first bump in an EKG?
Atrial depolarization.
Yes, this is when the atria are depolarizing, the little P wave. And what's this next big thing?
Ventricular depolarization.
This is when the ventricles depolarize. The QRS. And the last little bump?
Ventricular repolarization.
When the ventricles are repolarizing, the PQRST. During this QRS, what valves are shut and which valves are open?
AV valves are shut and the pulmonic and aortic valves are open.
Yeah, so this is ventricular contraction. So when it's pushing back up at the atria, you need to make sure the AV valves are going to be shut so you don't squish blood back up into the atria. And at the very beginning, the semilunar valves will still be closed until you have that isovolumetric contraction where you have to overcome diastolic pressure. But once you do that and achieve ventricular ejection, then the semilunar valves are going to have to be open to allow the blood to leave the ventricle towards the lungs. What else did we do here? We looked at the pulse wave. We looked at how fast, what's the velocity? Like, here's a person. This person, their heart sends blood out the aorta, goes down all the way to the finger. We wanted to see, once the pulse wave starts at the heart and then shows up at the finger, we wanted to see how fast. How did we calculate, how did we figure out how fast that pulse wave goes as it travels from the heart along the brachial and radial arteries to get to the finger?
Speed is distance over time. So we measured the distance using a ruler, and then the time, we used the power lab for that. We looked at the difference between the QRS and when the pulse arrived at the finger.
Yeah, exactly. Remember that? We actually could tell the moment of ventricular contraction by the EKG. And that's that force that's creating the pulse wave initially. But then we also had that little transducer that we could see when did the finger actually swell out, when did the pulse wave hit. We could measure that delta T. And then you could just use a tape measure to measure what's the physical distance. And then it's just a simple equation. So that was pretty easy. What else? Then we did a few things that were about the respiratory system. We did spirometry. We did that exploring the respiratory system. This one, how is exhaled air different from the air that you originally breathed in?
It's warmer and has moisture.
It's warmer and moisture. It had actually one other thing. What else? How else was it different?
CO2.
Or CO2. But I mentioned that it's important to like actually take note. The fact that it's warmer and moister, why is that worth paying attention to?
Can dehydrate more if you're blowing off more water.
Or, I mean, dehydrate more. It just means that the very act of breathing is one of the ways that water is leaving your body. So it was a important part of that insensible water loss, right? This idea that no matter how hard your body is trying to conserve water, you are always losing at least like a liter of water a day as part of this insensible water loss that you can't no matter you can tell your kidneys hold on to more water with ADH and this and that, but just every time you breathe out, there's a little bit of moisture leaving you, and that's part of the kind of debit side of your water balance. And again, also your energy balance. You're cool. You're losing energy every time you breathe out, and you've warmed up the air a bit. You're losing energy. We did the thing which you guys did at home, which hopefully was kind of fun, where you breathed at different ways and try to hold your breath. What was the way to hold your breath for the very longest?
With hyperventilation.
You did that hyperventilation, and that was how you could really hold your breath. Why does that make sense?
You're getting rid of more CO2, so it's going to take longer when you hold your breath for that CO2 to hit the critical level.
Exactly, because it all comes down to the fact that your urge to breathe is driven by CO2 levels rather than oxygen levels, right? So when you blew off all that CO2, it's down low. It means it's going to take longer to get up there. But anyway, the spirometry. You should definitely know the basics of spirometry. What do we call it? If this is time, this is volume in liters. If I'm just breathing, what do I call this volume with just quiet breathing?
Tidal volume.
That's that tidal volume, or V sub T, or and again, about a half a liter. If I breathe in the most I can above a normal inhale, what do I call that?
The inspiratory reserve volume.
That inspiratory reserve, right? That IRV. Or I could blow out the most I can beyond a normal exhale. The expiratory reserve. Again, the sum total of all of that, IRV plus tidal volume plus expiratory reserve, what do we call that total volume of exchangeable air?
Capacity.
Vital capacity.
Yeah, exactly. And again, remember all of these are positive numbers, right? So an IRV is typically around 3, tidal volume is typically around a half, ERV is typically around 1 liter. So in terms of the volumes, we just use positive numbers. So we'd say like a typical vital capacity is somewhere around 4.5 liters or something here. Then we talked about the forced vital capacity, where you just breathe in and breathe out as most as you can, all in one big go. Hold on, I'm sorry, I have to run 1 second. So this forced vital capacity, which was this thing we used for this kind of clinical clinical assessment of kind of respiratory health. We looked at how much of that total amount could they get rid of in 1 second. Well, how do you use that to think about their respiratory health? Like we looked at, we call it the FEV1. How much of that volume did they get out in the 1st second compared to the total amount? This was the FVC. This was the FVC in 1 second. How do you interpret this?
It was a ratio of FEV over FVC.
Right, and so yeah, I mean this is a ratio, FEV1 over FVC. What's that percentage or proportion? And how do you interpret it though? What does it tell you?
If it's below 80 or 70, then you might have some pathology or like constriction in the lungs.
Right, so typically you should be able to get at least 70 or 80% of that total FVC out in the 1st second, assuming everything is okay. If it's not, that's usually a sign of some kind of restrictive pulmonary problems where you're having excess resistance, and even when you're trying your hardest to blow it out as fast as you can, you're not being able to. Like asthma or something, where you have constriction and increased resistance, right? We looked at coughs. How is a cough different from a regular exhale?
Higher velocity.
Yeah, we saw it was a higher velocity. How about the amount of air that left in a cough?
Less than?
It's the same.
About the same. It was not much different than a normal, like, quiet exhale. But we saw, hopefully you have that in your book. We saw that quite an increase in outflow velocity, but really not that much. And why is that useful? Why is it good to have like high velocity but not such an increase in volume?
If you continue to cough, that you still have.
Yeah, exactly. So you've
Still.
got that impulse. You've got like that really that kind of forceful thing that comes out, but you still got air left over. So assuming that you need to do another cough, you can. Right? You can keep going. The last ones we did, and I'm realizing it's 12:30 now. I should do. The last two were the urinalysis, the kidney lab regulation of osmolarity, and the digestive lab. Urinalysis, you should just be able to know what kind of things you're testing for and what they mean. The kidney lab, for those 4 different groups, you should kind of understand our results and why they make sense. And the digestive lab, the one thing we kind of rushed at the end of this, what did people find happened in the protein part of the lab? Did anybody actually finish this up? Hopefully you have.
Yes, I did, but I didn't see any video, Professor, where I could see if the egg was dissolved. So I predicted that if in process of pepsin, I don't know what I wrote, but it was not in the video.
Yeah, you left that part. You left those results out of the video.
Did I not even have the video?
No, you had the video, but you left the results for proteins for the egg whites. You guys never showed the tubes at the end after waiting an hour.
We just showed the procedure and how to do it.
I thought you said because it was
like really crappy results and like not really
Oh, yeah,
worth
we
doing.
basically got crappy results on it.
Uh-huh.
So, right. So on that one, but all the other ones we got pretty nice results. Like what was the best if you really want to break down fats, what would you want to have?
Add biofads.
And
Pancreatin.
And the pancreatin.
right, because bile salts on their own emulsify but aren't going to actually break down, but together they
And
help.
pancreatin, yes.
How did you know if fats was breaking down?
Turned pink. The litmus cream turned pink.
Yeah, that litmus cream, because and why is that a sign? How does that work?
There's more acidity from the breakdown of the fats into fatty acids.
Yeah, exactly. Exactly. Um, and also for the carbohydrate one, make sure what were the two things we tested for when we're looking at the carbohydrate breakdown?
Starch and
The Lugol's and Benedict's test.
And
So like
maltose.
if I had starch and saliva and they sat for like 1 hour, and then I checked for if I did my Lugol's test and I did my Benedict's test, what would they look like?
The Lugol's would be dark purple, and the Benedict's would be blue.
Why?
Because if it turns dark purple in the Lugol's, that means that there's starch present. And then if there isn't any maltose present, then it'll be blue for the Benedict's test.
So let's think about what are you going to expect if you have.
Oh, never mind. There will be maltose present if it's saliva and starch. So it'll be like a reddish color in the Benedict's.
So Benedict's is going to be positive, so not blue. Again, it can even be green. It can be lots of different colors, but it's not going to be blue. What would the Lugol's look like if you've had starch and saliva sitting for a long time?
An amber color.
No, it'd be dark purple. Oh,
no, blue, dark purple. It's positive.
But I thought
Presence
that
of starch, no?
starch present was dark
If
purple.
starch and saliva are sitting together for a really long time, what's going to happen to the starch?
You'll end up with maltose.
It'll break down, so it will not be.
Saliva
the
breaks down starch.
Right, the starch is going to break down. There's not going to be any starch left after a while, right?
So wouldn't it be the other color, the amber color, like iodine?
Yeah, exactly. It's going to be, right? What if I put just starch with H2O and that just starch and H2O sitting for an hour? What would happen?
Nothing would happen. It would
What
still
would
be
these
starch.
be?
It'd be the dark purple black color.
Right, so this is going to look more kind of this dark purple blue or whatever. It's more dark purple black. What would the Benedict's look like?
Blue.
Yeah, this is just going to end up staying kind of blue because we're not going to have any maltose showing up, right? So make sure you've kind of got in your head like what happens and how do we assess for it.
Professor, I have a question. I was a little confused. It's wording. When Lugol solution, it's for starch, no? So if it's blue, dark blue, it is positive for Lugol's. But if it stays in the Lugol solution a longer time, so it will start breaking into maltose, for example, it will be negative. No.
Oh, wait. But if it's so say it again.
Basically, if
When
there's
it's
starch there, it will.
when it's a starch, it's positive for Lugol's.
Yes.
So which color will be?
It's that kind of purple, that dark purple.
And when it turns to reddish amber.
So if it's reddish or amber, that means there is no Lugol. This Lugol's iodine on its own is kind of a amber color. So if there is no starch, it's just going to stay amber. But if starch is present, it will turn colors and get dark. So kind of similar like Benedict's. Benedict's is going to be blue If there's no sugar, that's just what it looks like. But in the presence of reducing sugars, it's gonna change colors and turn kind of orange or red or whatever.
Yes, I had it bad. I reading. That's why I'm asking. I was so confused about what means positive at Lugol's and negative, but I already understood.
Okay, good. Um, so are there other questions about these labs, or what's going on for the lab exam?
Can we briefly go over the results for the protein since we didn't have the results, but
Right. So
The
what we
results
did-
are there. There, it's like a Excel sheet or like a little
right.
thing.
But
So
as far as
let
just
me
kind of
take a look here.
explaining what the
I'll
possible
open it up right now. Digestion lab results. Um, wait one sec. Here it is. Opening.
like for example, for test tube 2 that had the hydrochloric acid, some of them had some results, but others had no
So
results.
here I go. I'm gonna share this thing right now. Share screen. Booby doop. So for the protein here, I have. All right, so this actually kind of showed they had reasonable, inconsistent, but at least results that were kind of made sense. Tube 3, what was in tube 3?
The pepsin hydrochloric acid, so that makes sense that had the best.
Right, so this would have the best one. This is the one that most closely mimicked what we found in the stomach here. You know, when we put, the ones that were just the egg white sitting in water by itself, nothing really happened. Egg white, pepsin, but in this really alkaline environment, that's so not what pepsin wants to be in. We didn't have any results here. Pepsin by itself, kind of, sort of. Hydrochloric acid, it shouldn't really do much, but here they wrote. But I would say the fact that tube 3 was the one that really had kind of more of a kind of consensus that there was some breakdown, that makes sense, given what we know about pepsin and its optimal pH that it likes to work in. So,
Right, yeah. I was more wondering like if I need to know more about the details for like test tube 2.
no, not
Okay.
really. Yeah.
Okay.
Um, so, any other comments or questions about this stuff? All right. So, and again, what I'm going to do my very best to kind of grade these labs and get you feedback on this stuff. So, you'll be better prepared to head into the lab exam.
Yes, I do have one question, Professor. But that last pepsin optimal function, it's with low pH, that's with hydrochloric HCl. But if it's there is not present of HCl, it will work still or it won't?
If at least from our data here, it's saying it's sure not working very well.
Okay. Okay, thank you.
Um, so, yeah, I mean, in general, that makes sense because for something that is used to or is optimized to work at pH 2, pH 7 is already pretty alkaline. And we talked about, pH scale is a logarithmic scale. So 5 differences, a difference of 7 to 2, that's 5 orders of magnitude. That's like 100,000 times as more concentrated H plus. So it's yeah, given that it's this exponential logarithmic scale, things that are separated by that much are actually very different. What else? So again, I have office hours until 1:30, so I'm happy to hang out or answer any questions or anything. Otherwise, I will see you on, I got this right now, Thursday.